When doing process development for complex products like cell and gene therapies, there are tens of thousands of potential options that producers have to choose from in order to create the optimal, most effective, and most cost-effective process. We've done some calculations in the context of cell and gene therapy, and just from commercially available products, there are over 19,000 different permutations of media, cell lines, and transfection reagents. If you were to actually evaluate all of those permutations at scales, it would actually take decades to get through and cost millions of dollars. What we have developed at Virica is really a technology stack that allows us to marry high throughput virology, which employs high throughput screening, as well as design of experiment, which is a statistical method that allows you to shrink the universe of possibilities to be able to test as many possible options as quickly as possible. So something that would now take you decades actually can take you as little as 6 months using our approach. The real value is a more efficient process to get the optimal yielding and lowest cost process. So we can optimize for yield, or we can optimize for cost, or we can optimize for a combination of those two very important aspects, which means being quicker to get to market and being more cost effective.